Hi, everyone. Welcome. Thanks for joining us today. We're just letting a few more people trickle in and we'll get going in just a couple of minutes or less, probably. Okay. So we'll go ahead and get started and uh, let folks jump in as they arrive. But welcome to today's Research Spotlight talk. We're broadcasting live from the Yale Peabody Museum, which, as many of you know, is undergoing renovations and nearing completion, which is exciting. So we'll be opening up to the public in April. So stay tuned for updates on the actual opening ceremonies. And a quick intro about myself. I'm Katrina Damrell. I am a Master's of Environmental Management student at the Yale School of the Environment. And I'm also the Research Spotlight Assistant at the Yale Peabody Museum. The Research Spotlight series is the Peabody's graduate student speaker series. We highlight all the different grad students who do the research in connection with our museum's collections. And so I have a few housekeeping details to run by before we get going. Since today's event is a Zoom webinar, we have disabled the chat function. So please use the Q&A button on the bottom of your screen if you have any questions. And you can submit them at any time during the webinar. It won't throw us off. And off screen here is my colleague, David. He's managing the camera and the Q&A. So he'll be able to check your questions as they come in. We do typically receive more questions than we have time for, so we apologize in advance if we don't have time to answer all of them, but we'll certainly pass along questions to Alex, who I'll introduce in a second. And you always feel free to reach out if you do have questions and ran out today. Also, we're being live captioned, and you can turn the captioning on in the menu on the bottom right-hand corner of your Zoom window. So thank you, Diane, for captioning for us today. And then finally, I'd like to introduce our speaker. So we're joined here today with Alex Bertacchi. Alex is a PhD candidate in Yale's Department of Anthropology, and he grew up in Tuscany, Italy. His work focuses on the behavioral ecology of foragers in Eastern Africa in the modern-day region of Malawi, with a lens on how climate can affect food and resource availability. His talk today will illuminate how fossilized teeth from plant-eating animals can help us learn about the climate and vegetation 20,000 years ago in the Lake Malawi region. Lake Malawi itself happens to be one of the deepest lakes in the world. And with that, I'll pass off to you, Alex.
Thank you, Katrina.
Yeah.
Okay, I'm going to share my screen. Okay, so thank you for inviting me. I'm really excited to share some of the things I've been working on, and specifically how we can learn something about ancient environments using the chemical composition of fossilized teeth. So this is the kind of material I work with. This is a modern Eastern African antelope. And so the first step is to collect some material, some tooth enamel, to read clues about the environment that are preserved in the enamel. So you might notice that the red arrow is pointing to a little scratch on the surface of that third molar. And that's the mark that is left after I remove some of the enamel for analysis. So you can see why these kind of analyses are called microdestructive. They use up a little bit of material, but very little. So they are also suitable for museum specimens or rare fossil specimens. So what do I mean by the chemical composition of the enamel? Well, most of the time, what we are looking at is the stable isotopes of carbon and oxygen. So isotopes are versions of an element that differ in the number of neutrons. So for instance, with carbon, we have carbon 12 and carbon 13. They are both carbon. They behave the same way most of the time, but carbon 13 has one more neutron than carbon 12, which makes it a little bit heavier. And why are we interested in how much carbon 13 there is relative to carbon 12? And we express this as difference from a standard. That's the Greek letter delta right there. That is because different plants have different photosynthetic pathways. And depending on the photosynthetic pathway, a little bit more or less of carbon 13 is incorporated in the plant, and the signal is passed on to the tissues of the animal that eats that plant. So in a tropical environment, lower delta carbon 13, that is lower abundance of carbon 13, is associated with leaf eating or browsing. Conversely, higher delta carbon 13, so more abundant heavy carbon, is associated with grazing or grass eating. So animals that are flexible in their diet will reflect the kind of plants that were present in the environment. Same concept with oxygen. In this case, we look at oxygen 18 relative to oxygen 16. And lower delta 18O indicates a wetter environment with more precipitation. And a higher delta 18O indicates a drier environment with less precipitation. That is because the lighter version of oxygen, oxygen 16, is more likely to evaporate from surface water or from a plant, therefore leaving behind in the surface water or in the plant a slightly heavier molecule of water. So as you can see, already with these two isotopic systems, we can tell some pretty important things about the environment, how wet it was and what plants were there. But there's more. We can collect multiple samples from each individual tooth. The tooth, as it grows, records environmental signals. And therefore, if we take multiple samples along the growth axis of the tooth, then we can reconstruct seasonal curves. In this case, the low parts of the curve represent the wet season, and the high points in the curve represent the dry season. So we can look at season length or how seasonal the environment was. The pictures on the sides represent show some of the specimens I've been working on. There are different types of animals, different degrees of preservation of the material. Some teeth are broken, some teeth are complete, some need to be mechanically cleaned before they can be sampled. But in all cases, at the end, as you can see, we have a band of clean enamel and multiple samples being collected along that enamel. But why are we interested specifically to do this in Malawi? There are a couple different reasons. The first reason is just paleoenvironmental interest. Malawi is located in southeastern Central Africa. It's not quite Eastern Africa. It's not quite Southern Africa. It's in between. And that means that it's not easy to infer paleoenvironment from records that are pertinent to Eastern Africa or to Southern Africa, because this environment is at the interface. And so we really need proxies or reconstructions that are specific to this region, especially because having such a big lake nearby influences the way the climate works. The map shows you where our study area is. That's the Kazitu Valley in the red box. And then the big picture shows the Kazitu Valley itself. That feature at the center is Mount Hora, which is a granitic Inselberg, a very prominent feature, still culturally important today, and it's where some of the sites that we have investigated are. The valley itself is at 1,400 meters above sea level, So it might look flat, but we are actually at elevation, and some parts can get higher than 1,700 meters above sea level. So it's a pretty rugged environment. The second reason why we're interested in this area is because we have independent evidence that sometimes around the last glacial maximum, sometimes around 22, 23,000 years ago, there was a major change in population connectivity. The first line of evidence that we have is material culture. The panel on the top shows some ostrich eggshell beads. We know from studies of modern hunter-gatherers that ostrich eggshell beads are exchanged to establish and reinforce social networks. So people trade them, and then the act of exchanging these beads means that they are partners, friends, allies, and so on. Miller and Wang have studied the ostrich eggshell beads going back 50,000 years before present, both in Eastern and Southern Africa, and they noticed that the styles are pretty much exactly the same, and it is extremely unlikely that that happened by chance. More likely, this means that people were in contact with each other over thousands of miles. However, after 33,000 years ago, the styles start to diverge, indicating that these people were not in contact with each other anymore. So something happened. The second line of evidence is ancient DNA, including ancient DNA that we recovered from our sites. This shows that if we go as far back as 50,000 years ago, people from Eastern, Central, and Southern Africa were all equally related to each other. So they were moving very long distances, exchanging mates, and being all part of one big group of people. However, after 17,000 years before present, we have extreme divergence in the DNA, suggesting that people, especially in Malawi and Zambia, were mating only over very short distances. So essentially, they were related only to people who lived within a few tens of kilometers around their sites. So these are very large changes, but what happened exactly? We don't know why these changes happened, but one hypothesis is that there was a change in ecological conditions, and specifically, maybe there were enough resources locally, and these were sufficiently predictable, that people might have decided to stop moving so much and stay local, perhaps even control smaller territories. So investigating the paleoenvironmental context and the distribution of resources around this time is what I set out to do. Let's look at some data. So we start with stable carbon. Towards the left, we have more leaf eating. Towards the right, we have more grass eating. And we have the colors represent time. So we have the last 22,000 years of conditions in the Kazito Valley represented. And I have highlighted two taxa, warthogs and small browsing antelopes. And what they show is that over time, carbon gets, delta carbon 13 gets lower, suggesting that there was more leaf eating in the case of the warthogs. And in the case of the small browsing antelopes, it was always leaf eating, but in the younger periods, so in the periods that are closer to the present, this happened in a more closed, more wooded kind of environment. So we go from grassier, more open environment to a more closed, wooded environment. If we look at the oxygen, we have a similar picture. This is a pretty complex picture, so I have simplified it by grouping all the types of animals together and just showing a time sequence here. And you can see that from oldest to youngest, we go from a drier environment towards a wetter environment. So it looks like the last glacial maximum was overall drier compared to conditions today. And here we have some of those intra-tooth sequences that I talked about. So here we have 4 wildebeest. A wildebeest is a large grass-eating antelope. And these 4 individuals have been directly dated with radiocarbon. So we know exactly how old they are. and they all date to the later part of the last glacial maximum. The orange lines represent stable carbon isotopes, that is the diet. And you can see that these lines are pretty flat. So the diet does not change throughout the year because that could affect other measurements. The blue lines represent oxygen isotopes. And you can see that we have these very big swings. We know they are big because we can compare this to modern animals that live under conditions that we can know independently. And that means that the environment was very seasonal. So there was a big difference between the dry season and the wet season. Presumably, this meant that the dry season was longer. And we know that these kind of conditions also result in an environment that is on average drier. So to reiterate the point, we know now that the last glacial maximum in northern Malawi was drier than today. The next step is, okay, we know that how the environment was, but what does that mean for the movement of the different animals over this landscape? And what did that in turn mean for the people who hunted these animals to eat? In order to investigate how animals use the environment, we need to use a different isotopic system, strontium, and specifically the ratio of strontium-87 relative to strontium-86. This ratio is used in forensics, in archaeology, in food science as a geolocator. Essentially, it can tell you where something comes from. That is because there is in the soil a pool of what's called bioavailable strontium, strontium that we can incorporate in our tissues. This pool enters the water system and it enters plants. And when animals drink that water and eat those plants, incorporate that environmental signal in their tissues. The bioavailable strontium, in turn, comes mostly from the weathering of the bedrock. So the substrate on which the animals are living erodes over time and releases in the soil strontium, some of which is bioavailable. So if we know how strontium changes over a landscape, then we can use that knowledge to trace where things come from. Okay, so we need a map that shows us how strontium changes over a landscape. And that's what's called an isoscape, which is a spatial model of how an isotopic ratio changes over space. In order to build an isoscape, we defined a study area, which in our case, it's about 2,500 square kilometers. Then we went out and we collected environmental samples, modern samples. So we went and we collected over 200 plant samples, both leaves and grasses. And we also collected dust samples and giant snails. And we sampled 3 types of environments. Non-riparian, that means far from a river. Riparian, that means along a river. And dambo. And a dambo is a waterlogged grassland. So it's a wet environment, but there isn't a river. The colored dots on the map shows you where we collected the samples. Okay, so now that we have all these samples, we can create a model of how strontium changes over space. There are many techniques to do that. I used what's called empirical Bayesian kriging, which is a technique that subsamples your group of environmental samples and creates a map and repeats this process many times, in this case 10,000 times, and then it creates a consensus map. And you have it here on the left. So this is still our study area, but the blue parts represent the lower ratios of strontium-87 to strontium-86 in the bioavailable pool, whereas the red parts represent areas where this ratio is higher. And then the map on the right represents the estimation of the standard error of the prediction. And this is assembled using a leave one out procedure where we leave one sample out and then we compare the predicted value at that location with the known value and we create this uncertainty map. The lighter colors in the right-hand map represent less uncertainty. And so you can see that we are more certain of the southeastern quadrant of our map. Okay, so now that we have the map, we can actually measure these strontium isotopes in the teeth. We have 51 teeth from 19 teeth and 51 samples. So it's about a quarter the size of our other data set, although we have about 100 more coming. And you can immediately see that different animals inhabited different places. On the top row, we have zebras. They are all the way on the left part of the graph. And on the second row, we have warthogs, and they are all the way on the right side of the graph. So there are some differences. And in some cases, within the same species, we have time differences. So animals were inhabiting different parts of the landscape at different times. So we could then match these ratios to the ratios in the map just visually. However, that's much more complicated than one might think because there is a lot of uncertainty built into the model. So a better way to do that is to use a mathematical model to create a map that shows you likely areas of origin for every individual animal. And this is what I've done. So here we have satellite images of the study area, and then overlaying on those satellite images, we have these blue pictures. And so the blue areas represent, the bluest the area, the more likely is that the animal came from there. And the blue pockets around specific sampling locations are a methodological artifact. So we are looking for broad areas of blue color. And you can see that in the case of these small browsing antelopes, they were mostly hunted next to the sites where they were found, except for the one on the right that has an intermediate value and could have come from anywhere. If we look at the zebras, we can see that last glacial maximum zebras on the left-hand side were foraging and presumably were hunted somewhere in the mountains, in the highlands, in the southeastern quadrant of the map, but far away from rivers. So people must have hunted them and carried them to the site where we found them. And we know from traces of butchery on their bones that people were exploiting them. If we look at this late Holocene zebra in the middle, you can see that it was hunted still in the southeastern quadrant of the map, but more likely along a river. So different places. And the warthogs instead might have come from a little bit anywhere except the southeastern quadrant of the map. So a completely different place. And then we can do this with other kinds of animals, such as, for instance, these large antelopes. And for some of these animals, we can exclude some of these possible areas of provenience based on what we know about their environment and their behavior. So some antelopes, like the one on the right, are more water dependent, and so we can exclude areas that are far away from the water. And so we can further constrain where they might have come from. Okay, so what did we learn from all this analysis then? So the first thing is that we know now from carbon and oxygen isotopes that the last glacial maximum and the terminal Pleistocene were more seasonal and overall drier than the Holocene, which is our time. We then know from strontium isotopes that small game was hunted locally, whereas large herbivores were hunted in the higher elevations to the southeast of the archaeological sites. Critically, we have no evidence of ungulate migration or long-distance hunting. So we can't prove that something was hunted locally. However, none of the animals that we tested had strontium ratios in their teeth that are incompatible with them living nearby the sites. And all this is consistent with the idea that people were leading a more territorial, less mobile life way. And with this, I'd like to thank all my many collaborators and co-authors, both in the field and in the lab, and also the funding bodies, especially the Yale Institute for Biospheric Studies and the National Science Foundation. And I'll take any questions.
Awesome. Woo-hoo. That was fascinating. Thank you so much, Alex.
Thank you.
Yeah. So we have a few questions. David was wondering, are the strontium ratios in leaves always a close match with the strontium ratios in the soil collected near those plants?
Yes, most of the time. So strontium in the soil can change with root depth, so with depth. And so plants with different root depths can have different values. However, that's why we collected both leaves and grasses. And in our study area, they have a relatively small difference. And so we can be pretty confident that at a certain location where if you measure leaves, grass, or soil, you're going to obtain roughly the same measurement.
Interesting. Huh. The strontium is a new thing I learned from your lecture too. It's really fascinating how that can be used as a biomarker in so many other aspects of science too. There's another question about whether do we know anything about the temperature differences in the same region between the last glacial maximum and present day? How?
Yeah, there are some reconstructions that are based on chemical biomarkers that are deposited at the bottom of the lake. So there are some cores that are extracted from Lake Malawi, and other people do all sorts of analysis on them. Some analyses are used to infer temperature, and the estimate is about 4 degrees colder during the last glacial maximum as an estimate of the yearly average. And that doesn't mean that it was cold. Like when we talk about the ice age, it's the ice age because the global glaciers were larger, but in the tropics it was still hot. So instead of having an average of maybe 24 to 26, it might have been 22 or something like that.
Right.
So and that is enough to make a difference. So yes, it was colder, but it was not cold. And part of the reason is because the concentration of carbon dioxide in the atmosphere was higher. And so it kind of shielded a bit the solar radiation.
Oh, interesting.
Yeah.
I thought that. Yeah, I have another question. What time, like when did you guys do all the field sampling and yeah, what has that aspect of your work been like?
Yeah, so it's pretty fun. It's a lot of hiking. So there is a team led by Professor Jessica Thompson here at Yale.
Who you had on your last slide, I think.
Yeah,
right?
she's my doctoral advisor, and they go out every year, every summer. Of course, there was a hiatus during COVID, and I have gone there in 2018 and 2019. Most of what happens is excavation, but on the side, I was able to get a field vehicle and with my collaborators for over a period of 10 days, drive around, collect plant samples or other types of samples, and just collect the location with a GPS unit, and then export the plants to the United States for analysis. And other people had done some sampling as well in 2018 and in 2017. So over 3 years, we collected all the samples. The first field collection was done somewhat randomly. It wasn't completely random, but it was essentially using a random number generator or something to create a first map. And then from there, we tried to either fill in the gaps in areas that were undersampled, or if there were areas where we weren't quite sure what was going on, we then targeted specifically areas that were quote unquote problematic.
Okay, because there's an existing collection from that region from your advisor's work.
Yeah,
like that's what you're.
Yeah, it was
a
postdoc at Illinois called Andrew Zipkin. He had started this in 2017, and then I took over in 2019. Yeah.
It's fascinating. We have a question from Vince. Was the seasonal movement of animals mentioned in terms of sampling and results?
Yeah, so so we don't know a priori if the animals were migrating or not. So
Okay.
obviously they move over a certain area. They have a home range, a daily space that they use. However, some of these animals might have migrated long distances, and therefore they would have been available locally only during a specific portion of the year.
Right.
And I would have expected, for instance, the zebras to migrate long distance, just based on because we know from zebras in national parks today in the area that they don't fare too well. And so they lose body condition during the dry season. So I thought, okay, if there were no fences, if these national parks were fenced, they would move to wetter areas during the dry season. But instead, in the strontium, we don't see that. The values that we get for the summer and for the winter are the same.
Oh.
So that suggests that they were staying locally year round.
Interesting.
Yeah.
So modern day zebras are also still more localized?
Yes, but that's because they don't have the capacity to move long distances because they are fenced in the national parks that are large, but not as large as they should be for them to be able to migrate.
Right. Yeah, okay, that makes sense. I had another question about when the sampling, when we were first looking over your presentation, you mentioned the sampling of the soils along the riparian and Dombo areas. And is there a goal, like I noticed that there was a different number between total amount between those and the drier, more higher elevated areas. Is that something that affects the results or?
Well, yeah, potentially, but there are many more samples from non-riparian environments just because in terms of surface, most of the land is not along rivers.
Yeah.
And we wanted to maintain a somewhat even coverage over the whole 2,500 square kilometers. And so most of those samples are by necessity going to be non-riparian.
Okay.
And then on top of that, we did collect samples along the rivers to target the riparian ones. And there is also a transect that we did that goes from Mount Hora to the closest river, and it collects like samples every few meters. But yeah, that was just by necessity.
Yeah, that makes complete sense. Field studies are on a timeline too. Yeah. I'm wondering more about the community around Malawi right now and the region that you guys are studying. Are there opportunities to engage with the community there in the research, or?
Yeah. So there are several levels. First of all, in order to do research in the first place, you need to obtain a permit from the country. Then we also obtain permission from the traditional authorities, so not the government, but the kind of the chiefs of the different areas where we work. And there's a hierarchical system with a paramount chief and then other chiefs. And we talk to them, we get their kind of agreement. Then in terms of the actual collaboration in the field, there are two levels. We have a set of collaborators who are always the same. We've been working with them for many years. They are all Malawians, and they travel to wherever the field season is that particular year. And there's like 7 or 8 of them. And they have kind of managerial roles. So they have a lot of experience, and so they for instance, the field lab is managed by our long-term collaborator Kondwani, and so on. And then the local communities, as in like the people who live in the immediate area, typically, as soon as we arrive, they ask to be hired to do different field tasks, which is great. So we employ maybe 20 to 30 people from the local community, and then some of them decide to come back year after year, and they sometimes learn specific skills. So there is that. Then in terms of outreach, we do some outreach with the local community, and sometimes we are involved in whatever cultural activity that is there, or if there is something that needs to be fixed, etc. At the moment in Malawi, there isn't an archaeology program in the university. There was one, but unfortunately it has closed. So there aren't people who get trained specifically in archaeology, but there are people who maybe are doing a degree in geology or history that are interested.
Like adjacent subjects. Yeah.
Yeah.
That's awesome. That's cool to hear about the longevity of all the work that you guys have been working on as a lab. Yeah. That's awesome. David is wondering, were most of the ancient human communities along rivers? And if so, how might that have influenced the variety of animals that were hunted?
Yeah, so they weren't along a river per se. They were always close to rivers, but the sites that we found are never like immediately adjacent to it. And that could also be just because the kind of sites that we are likely to find are not very close to the rivers. Usually what we find when we survey is a big boulder that forms an overhang. And we go under it and we see eroding down the slope some artifacts or some bones, like just being scattered. And then we follow them up from where they come from, and we find them eroding, and then that's where we target for excavation. And these big overhangs protect the site from rain and erosion. They can still get eroded because especially termites can destroy everything. But if there was a site that was just in the open, far from any like durable feature, and then now it's buried under 2 meters of sediment, we have no way of knowing it's there. For later time periods, when you have walls and other big structures, you can use geophysical techniques to like see through the terrain, like with a radar, for instance.
Yeah.
But in our case, these are just scatters of artifacts and bones.
Because it's so old, or
yeah, they just didn't. They just didn't build walls.
Yeah.
They probably did build structures made of plant material, but that doesn't preserve. So we usually just find a specific type of site, which is not any site that has existed.
Yeah.
In terms of what animals were around, yes, that definitely affects the kind of animals that they were hunting. However, there are 2 levels to it. There are the animals that are immediately around the site that they most likely hunted upon encounter, as we kind of say in jargon, which means they just bump into them, decide to initiate a hunt, or perhaps they put some traps or other things like that. And then there are other animals, usually very large animals, where people would specifically walk to the place where they are to hunt them because they are so profitable. They have so much meat.
Yeah.
And so, for instance, in our map, that southeastern sector where a lot of the big stuff seems to come from, that's just 20 to 30 kilometers from the sites, provided it's a pretty rough terrain. There is like, you're going up and down, and etc. So it's not as the crow flies. This being said, by hunter-gatherer standards, we are still calling this local, right? So like long distance interactions are at the scale of the hundreds of kilometers. There must be, for something to be long distance, you need at least 1 order of magnitude more in terms of distance.
Wow.
And this is just like kind of disciplinary convention. There is no like intrinsic reason why it should be that way, but
right. Just kind of the rule. Yeah. Awesome. Well, I think about that about wraps up our
right.
time with you today, Alex. But thank you so much for sharing all of your research, and congratulations on wrapping up your PhD here.
Thank you. Thank you for having me. It was great.
Yeah,
Yeah, very fun.
And before we sign off, I just want to let everybody know that the Research Spotlight series will continue, and later in the spring, on May 16th, we will be having Caleb Gordon from the Department of Earth and Planetary Sciences join us, and that will actually be most likely in person. So we're very excited about that opportunity, and stay tuned for more information on that. A great way to do that is to follow Yale Peabody Museum on social media or sign up for our newsletter. Awesome. Well, thank you again,
Yeah, thank
Alex,
you.
for joining, and thanks, David and Diane. Off screen. See you guys.
Bye.